But something as unique as each and every one of us that doesn't interfere with the well-being of other species. Greetings, my name is Marisela Lapetoro. I'm a high schooler from Petra Mercado Bugar in Macau, and my project consists of the development of a new era, personalized medicine and stem cells. When I first wanted to create this research, I asked myself, is it possible to create medicine based on our own genomes while replacing animal testing with stem cells? I then created a hypothesis that made me think that by implementing this new approach, it'd bring an end to what seems like a never-ending cycle of unnecessary drug use as well as animal abuse. Over 100 million animals get killed due to animal testing, and that's without taking into effect all of the abuse that they endure when conscious. Researchers force them to eat, inhale, or inject the substances to see their reactions. However, animals tend to get high levels of stress caused by the hearing of other animals suffer, sometimes even their own family members, and this causes them to possibly mutilate themselves. The biggest funder of research in the US, the National Institute of Health, spends nearly half its annual budget on animal studies that 90% of the time fail to provide a treatment for humans. That's $19.6 billion down the drain every year. There is a solution to all of this, which you can observe in figure number 1, timeline of the genome era, which is how we discovered the DNA, and from there on went to the genes, and from the genes to the genome era. To read this research, I first searched on PubMed, Google Scholar, and JSTOR databases, which we can observe in figure number 2. I then recompiled all the necessary information and developed questions about the topic. Restrictions were made in the 2018-2022 timeline, along with keywords such as genomic sequences, animal testing alternatives, personalized medicine, stem cells, and drug development. That led me to establish a hypothesis that made me analyze the problem, search for resources to provide, and justify my answers to conclude the findings. It started from no testing to animal testing, but we can take a much safer and healthier approach. As a result, this new alternative would be isolating the patient's stem cells and treating them with the reprogramming factors, as we can observe in figure number 3, until they're able to produce a variety of cell types, in which you could test the safety and efficiency of the drug in development before giving it to the patient. A better example for the past image would be targeting cancer cells, in which we can use immunotherapy and pathway-based targeted treatment that submits the patient to a tumor cell extraction that leads to tumor biomarker studies. Then they can decide the perfect fit for the patient, whether it be with a new drug in development or taking more tests while getting a closer look in therapy. If it were to be a new drug in development, instead of it being tested on animals as per usual, it'd be tested on the patient's own stem cells. I list the top of my 5 references. Thank you so much for your attention. Are there any questions?